What happens when we consume proteins in our diet? During digestion, those proteins are generally broken down into their individual amino acid units. The way that this happens is through a type of reaction referred to as a hydrolysis reaction. Hydrolysis refers to the breakage or lysis of chemical bonds using water, hydro, as a reactant. So in this video, we are going to look at the specific type of chemical reaction that breaks the bonds between individual amino acids during digestion. We are not going to focus on the enzymes that catalyze these reactions. Instead, we'll save that for the next video. So in this video, we focus exclusively on the chemical reaction known as hydrolysis to allow us to break the amide linkages, also known as the peptide bonds, between amino acids within a protein. The digestion of proteins start with intact three-dimensional structures, that is, the tertiary and quaternary structures of the protein that are ingested in our food sources. Through the process known as denaturation, which we covered in the last video, these protein three-dimensional structures are subjected to the acidic environment of the stomach, and that acidic environment enables the protein three-dimensional structure to be disrupted, resulting in the formation of just a disorganized polypeptide chain. By creating this disorder in the protein, that will more readily enable enzymes generally known as proteases to come in and catalyze the breakage of the amide linkages, also known as the peptide bonds, between the individual amino acids. We will focus on specific proteases more in the next video. Here we're focusing just on the chemical reaction. What happens when those peptide bonds are broken is that this polypeptide chain of many amino acids linked together covalently is broken into simpler and simpler peptides until ultimately we generate tripeptides, dipeptides, and other units that are much more simplified. And that's why I have multiple reaction arrows here and here indicating that it's not just one chemical reaction that takes a very long polypeptide chain and breaks it into tripeptides or dipeptides. Instead, it's several reactions going on, and that's why there are multiple arrows here. Those several reactions that go on, though, all follow the exact same pattern of breaking peptide bonds. Ultimately, then, through the action of enzymes, these smaller peptide units become broken completely down into the individual amino acids. And this is where, at this point, these individual amino acids can then be used for anabolic processes or processes that build up complexity, meaning that these individual amino acids, under the directions of your genetic code, will be reassembled into the proteins that your body needs. Now let's take a closer look at the hydrolysis reaction that breaks the peptide or amide linkages within proteins in order to ultimately release the individual amino acids. This reaction should look familiar because we did cover this reaction in an earlier video. So we'll review it here, and then in a moment we'll apply this reaction toward the specific scenario of dipeptides, tripeptides, and so forth, looking at how bonds can be broken in those. In this reaction, we take our amide starting material, reacted with water. Keep in mind our starting material is classified as an amide because of the fact that we observe a nitrogen directly bonded to a carbonyl. This is the group that links the individual amino acids together in a protein. When this reacts with water, generally catalyzed by an enzyme, the water is going to come in. The water oxygen will attack the carbonyl carbon here and bring a hydrogen along with it to ultimately result in a COH group forming here. This will cause the breakage ultimately of this carbon-nitrogen bond, releasing the nitrogen here, which picks up the other hydrogen atom from water to give an additional hydrogen here and keep its complete octet. Let's draw out the products of this reaction. Looking at this reaction, we see that our R groups here are defined as carbons and or hydrogens. I'm going to continue using those R designations and color code this the same in the products as I did in the reactants. So as we go through and we break this carbon-nitrogen bond with the OH group coming in and forming a bond here to the carbonyl carbon, that is going to result in keeping the black R group still bonded to the carbonyl carbon because nothing has happened to that carbon-carbon or carbon-hydrogen bond there, shown as the bond between carbon and R here and here. This carbon, though, is no longer going to be part of an amide group. Instead, it will form a covalent bond to the OH group of water. And therefore, I'm going to go ahead and draw in blue to match the color of the OH group from water on our reactant side. Here on the product side, to illustrate this OH group was donated by water. So now we have our carboxylic acid group COOH here. We've broken again that bond between carbon and nitrogen here on the reactant side in our amide, and the nitrogen will pick up a proton, or hydrogen atom as you might call it, from the water to have a complete octet on it in the product. So let's go ahead and draw the structure of that as well. So we have our nitrogen atom, which I will draw in blue here. Still has that lone pair of electrons because we haven't done anything to that. Still has R in red, which could be a carbon atom or a hydrogen. We still have R prime here, which could again be a carbon or a hydrogen, doesn't matter. And then I'm going to put in blue to match the blue color of one of these hydrogen atoms from water, a hydrogen atom directly bonded to nitrogen in place of what was originally the bond between nitrogen and the carbonyl. Now becomes a bond between nitrogen and hydrogen so that this nitrogen can still have its complete octet. So effectively in this reaction we have broken or lysed the bond between carbon and nitrogen to result in the formation of two products from one organic reactant. Hence, we will take a relatively complex protein that has amide linkages, and by breaking those amide linkages, be able to finally release individual amino acids via this hydrolysis reaction. In this problem, we are asked to draw the products resulting from hydrolysis of the following peptide, remembering that hydrolysis is the use of water to break the amide linkages, also known as the peptide bonds, within this peptide. We could do this exercise for a peptide ranging from anywhere between 2 amino acid units linked together and 2,000. In this particular scenario, we can determine how many individual amino acids make up this peptide by looking for the number of amide linkages, keeping in mind that our individual amino acids, as we learned in an earlier video, are joined by amide linkages. Looking for the amides within this molecule, we see an amide linkage here, remembering that that is a nitrogen atom directly bonded to a carbonyl. We see another over here in the backbone of the molecule, and those are the only two that I see. So with that in mind, and keeping in mind that the formation of the peptide resulted from linking a carbonyl group of a carboxylic acid to the nitrogen of an amine, we have a amino acid that we can make as a product here, one in the middle, and one over here. So in other words, this would represent a tripeptide, where we have 2 peptide bonds that are linking in total 3 amino acids. Now, when we do the hydrolysis reaction for this, what happens is that each of these amide linkages here and here will be broken using water as the reactant coming in. When we bring in water as the reactant, like we saw just a moment ago with our general reaction for amides, we break this bond, the carbonyl group becomes bonded to an OH group to make a carboxylic acid group here, and then over here, the nitrogen becomes bonded to one of the hydrogen atoms from H2O to create an NH2 group here at this structure's end. We would do the same thing over here. So let's go ahead and draw out the products resulting from breaking each of these amide linkages using water in this hydrolysis reaction. I will start by redrawing the molecule from the left-hand end here, the N-terminus as we could call it, because it has that free unreacted amine group. And we'll go ahead and draw that out as NH3+. We come here and we come to the alpha carbon right here that's adjacent to the carbonyl. That's where the R group hangs off. This has a methyl group hanging off of there. And if you compared that to your amino acid chart, you would see that R group represents an alanine residue. We come over to here and we're still bonded to the carbonyl, so I'm going to go ahead and draw in my carbonyl carbon, like so. I will also go ahead and highlight that in pink since it was pink to start with. And then we've broken the bond between our carbonyl group and nitrogen, and remember that the carbonyl group is going to become bonded to an OH group donated by water. And so I'm going to put that in blue, representing the hydroxy group donated by water, much like the blue we used over here. Now that this bond is completely broken, this molecule ends with OH. We'll start the next molecule, the next product of the reaction, with this nitrogen here. The nitrogen, to have a complete octet in the product, has picked up a proton from water, so it's going to become NH2. Let's go ahead and draw that out. So I'll go ahead and draw out NH2. And that NH2 nitrogen originated from our starting material there that was highlighted in pink. So I've highlighted the nitrogen in pink here as well. That nitrogen is still bonded to the carbon at the alpha position that I'm highlighting here on the reactant side. So I need to go ahead and draw in that bond to our carbon atom. I'll go ahead and draw that in like so. And then coming off of there, we have our R group. This is the R group for phenylalanine because of a CH2 followed by an aromatic ring. So I'm going to go ahead and draw that in over on our product side. We had our CH2 and then our aromatic ring there. And then continuing onward from our alpha carbon, we come to our carbonyl carbon, which would be right here. I will go ahead and draw that in. Carbonyl group. I will highlight that in pink as it was in the starting material, like so. And then that carbonyl carbon is directly bonded to an OH group, and so I will plug that OH group in. And that OH group was donated from water because we used water to break this bond that I'm showing here on the reactant side to ultimately give the COOH over here that we see on the product side. Now, following on through, our next place that we need to take care of is right here at this nitrogen atom that I'm highlighting on the reactant side. That nitrogen is going to be present in the product as NH2. The second hydrogen atom there was donated from water. So that nitrogen then bonded to not one hydrogen, but now two hydrogens. The nitrogen is bonded to the alpha position right here. And that alpha position is bonded to an isopropyl group. So go ahead and draw over to our alpha position. Draw an isopropyl group off of the alpha position. And then we're going to do COO minus to represent the other terminal of the molecule. So here we had COO minus, nothing's happened, so that remains COO minus. And with that, we have broken our tripeptide into the 3 individual amino acids. When we are thinking about the protonation state of these, meaning whether the nitrogens have positive charges and oxygens have negative charges, or whether those are shown as OH with no formal charge on the oxygen and NH2 with no charge on the nitrogen, it's usually best to keep that consistent in the products as well. And so here, for example, rather than showing the nitrogen terminal with a positive charge and OH here, it would be more reasonable to go ahead and clean up the final product to show O minus here, meaning since we have the Zwitter ion form at the left end, we should really make that a Zwitter ion on the right end as well to cancel that out. Here, if we assume that all 3 of these are in the same pH conditions, then generally the same protonation state is going to be favored. So if we did a Zwitter ion here, we should really do it here and here as well, cleaning those up to show it as the Zwitter ion. NH2, in order to become the Zwitter ion, that basic amine picks up a proton, so it becomes NH3 plus. And then at the other end of that particular molecule, we had OH. OH gives up that proton to the amine group to become O minus. So I'll go ahead and draw in our negative charge here, like so, and then do the same thing down here at the bottom. We had O minus at this terminal, and that would suggest that we need NH3 here at the left-hand end to create the Zwitter ion. So in other words, if one side of your amino acid is charged, the other side generally should be as well, because in a particular physiological pH condition, generally having a positive charge at this end will correspond to a negative charge at the other end, and vice versa, to cancel those out, making these Zwitter ion forms. Now, with this information in mind about the general reaction that is taking place here, we are ready to take a look at the next video where we dive into some of the specific enzymes involved in digestion that catalyze these reactions. Because while we could simply take water and mix it with a peptide and eventually yield these products, the reaction would occur very, very, very slowly. So slowly that it would not really be conducive to life. As a result, and are catalysts for these reactions that dramatically increase the rate of reaction and allow the body to exert control over metabolic processes.